Hello, everyone. Welcome to the last chapter that we will be going through for Bio 137. Chapter 14 is all about the brain. Definitely a subject that's near and dear to my heart. Most students do find this topic to be pretty interesting, so hopefully you will also find it to be so. Of course, the brain is a very broad topic. There is a lot we could discuss, but we have to narrow it down. So this PowerPoint, I think, is 122 slides, something like that. But really, a lot of it is pictures. A lot of it has to do with anatomy that you've already dealt with in lab. So I'm not going to dwell too much on the anatomy aspect. I'll just maybe mention a few things as we go. All right, so really a big focus for you all for chapter 14 will be the functions of the different brain regions. So that's really the big deal about this chapter. Most of your questions will come from that sort of material. All right, okay, so let's get started then. All righty, chapter 14, the brain. Right. So as an introduction, yeah, of course, human brain is extremely complex. It is, in my opinion, I think there's nothing more complicated than the universe itself. It's an extremely complicated part of the body to study. I would say that the brain, along with the immune system, would be the most complicated parts of the human body to study and to understand. So there really is a lot that we don't know about the brain. There used to be a saying, I don't think people say this so much anymore because it's not true, but there used to be a saying, you only use 10% of your brain. You only use 10%. Well, I'm here to tell you that is absolutely false. I think that statement came from someone who maybe indicated that really we only understand about 10% of the brain. And now that's true. There's still another 80 to 90% of brain function that we just don't understand it yet. We don't have it all detailed out. We may have some ideas about it, but so much about brain function is still under investigation. And this is because there are so many neural networks that are really complicated to trace and to understand, as well as the fact that the brain is probably the most individualistic part of the human body. So everybody's brain is different. Yeah, we have the same brain regions, but our neural networks are different because, well, obviously we all have different experiences. We may have similar experiences, but nobody has the same exact experience, the same memories, the same thinking. It's just going to vary so much amongst individuals. So this is another way that the brain, or another reason that the brain is so complicated to understand. Okay, so keep in mind, we use 100% of our brain. We use at least 100%. If there's more than 100%, we're going to use it. All right, or else it wouldn't be there. Why would all that tissue be there if it's not being used? That's not accurate. All right. And yeah, brain function really is associated clinically with the difference between being alive or dead. All right. And this comment down here, yeah, evolution of the central nervous system shows that the spinal cord has changed very little. And this makes sense, right? The brain has changed a great deal in comparison. And yeah, this makes sense because the spinal cord is really just a mechanistic thing. It's really strictly anatomy. There's no thinking going on, right? There are no memories there's no cognition going on there. There's no cortex, there's no consciousness, there's no thinking going on there. It's strictly an anatomical structure, the spinal cord. So yeah, it's changed very little over time, but the brain has changed a great deal, all right, a great deal. The greatest growth in the brain has been seen in areas associated with vision, memory, of course, and motor control of the hand, all right, specifically of the hand. Okay, so some terminology. The words rostral and caudal are typically words, directional terminologies that we're going to use strictly with the brain. I mean, you can use these terms in other areas, but predominantly rostral and caudal we're going to use in reference to the brain, sometimes the spinal cord, but and it's just because of the way the brain is situated, okay? So the front of the brain is considered the rostral part. The caudal part is referring to the back of the brain, which means closer to the spinal cord. All right, so the closer the brain, part of a brain to the spinal cord, this is considered more caudal. Caudal comes from the word tail. Now there are four main large regions of the brain, and we're going to break these down. Okay, so the first major part is the cerebrum. And this is really the cerebral hemispheres and the cortex, okay? So the cerebral hemispheres, it's really like the whole outside part that you can see from the outside. And you have two hemispheres. You have a right and a left hemisphere. Now, surrounding the cerebral hemispheres all around is what we call cortex. Cortex. Cortex is the most superficial part of the whole entire outside of the cerebrum. All right, and the cortex is a very interesting area of study. This is where you could say intelligence is located. Cortex varies in size amongst different species. All right, so the reason cortex is correlated with level or degree of intelligence is because, well, the human, all right, humans have more cortex compared to any other species. Whereas if you look at something like a sheep or a rat, okay, or a mouse, something sort of lower on the evolutionary scale compared to humans, you will find that they have less cortex. If you look at animals that people consider to be pretty intelligent, like dolphins, for example, or elephants, these animals are going to have more cortex compared to other animals on a lower level of the evolutionary scale. If you look at non-human primates, okay, monkeys, apes, they have a lot more cortex compared to other lower animals on the evolutionary scale. So anyway, the degree of cortex is correlated with the degree of intelligence of a species. All right, next major part is the diencephalon. The diencephalon really is kind of like the middle part of the brain. So if you go deeper into the brain, you're looking really at like some of the structures that are sort of in the middle. And so, you know, the structures that you know about would be like the thalamus, the hypothalamus, and there are others, of course. But yeah, you can consider thalamus and hypothalamus part of the diencephalon. Third main region is going to be the brainstem. And you saw this part in lab as well. The brainstem contains what's known as the midbrain. Part of the midbrain, we'll talk about a few parts, specific areas of the midbrain. You're already familiar with the superior colliculi and the inferior colliculi. These are considered part of the midbrain, and we'll also talk about some other structures there. The pons you know about, and the medulla oblongata, which runs into the spinal cord, right? So this is the brainstem. Remember that your cranial nerves branch from the pons and the medulla. And then the last major part, it's just an area all its own. It is called the cerebellum. Surface anatomy of the brain, okay, just a reminder of your anatomy, and you already know this. All right, we did this in lab already. All right, you've got your gyri, which are the like elevations. And then you have your sulci, which you have a variety of sulci. You will not have to know all of these sulci. You do need to know the central sulcus, all right, central sulcus. The lateral sulcus. Lateral sulcus is going to really define the barrier or difference between the temporal lobe and the parietal lobes. Cerebrum. Remember, the cerebrum is really this whole part, okay? And then you've got your cerebellum, which is all its own part here. and then your brain stem. Again, just another picture. Like I said, a lot of this PowerPoint is really just pictures of anatomy. So you feel, have a pretty strong foundation for the basic anatomy here. All right, diencephalon. So like I said, the diencephalon is sort of like if you go deeper in the brain, these parts that are here in the middle. Diencephalon, okay? So diencephalon, it's showing here, this is the thalamus. And then just below the thalamus, you have this very important hypothalamus, which we'll study a lot about in Bio 139. Interestingly, developmentally speaking, the brain actually develops in an inside to outside fashion. So, and this correlates with what are the most primal parts of the brain, meaning the parts of the brain that are associated with critical functions, especially dealing with instincts, instinctual drives, okay? So we develop those areas first, and then the brain develops from an inside to outside fashion, so that the last parts of the brain to develop are going to be the cerebral hemispheres and the cortex. The cortex is the last area to develop, just if you're interested in that. All right, brain stem, right? We've got midbrain up here. You can sort of see these colliculi here. You can see the superior colliculi and the inferior colliculi there. The pons and medulla. All right, again, just another look here at the brain, the superior view here. Central sulcus, you can see. Here's a mid-sagittal view, and you've seen this in lab before, so I'm really not going to go through and review all these with you. You really should know these at this point. Okay, again, here is a real human brain. If you want to look at that. Gray matter and white matter. I think I distinguished between these two in lab, but I'll go ahead and mention them again. So gray matter, the reason the tissue of the brain that is called gray matter is because it does not have myelin or very little myelin. So you know where myelin is. Myelin exists around axons. Okay, so this tells you that wherever there is gray matter, this is not where you find axons. Instead, it's going to be the unmyelinated parts of the neuron that are going to make up this gray matter tissue. All right, so gray matter then consists of somas, dendrites, and the synapses. Now, where do you find gray matter? You're going to find it on the outer layer, the surface, okay, the cortex. Okay, so this tells you that, the kind of information or the part of information processing that's occurring in the cortex is going to be where we're either sending information or receiving information. Okay, the final version of information, okay, the final version goes to the cortex. Okay, this is why cortex is associated with intelligence. Part of the reason is because, well, this is where a lot of our information is stopping. It's ending there. And this is where our final decisions are going to be made. Vice versa, this is also where our initial decisions or initial controls will be initiated here at the dendrites, okay, at the cortex. We also find gray matter deep in the brain in what we call nuclei. Now, when we say nuclei, in this sense, we're not talking about a nucleus of a cell. Okay, this is a completely different entity here. All right, when we say nuclei, we're just talking about the nuclei is referring to in the brain is referring to a part that's going to be unmyelinated. Okay, it's called a nucleus. Okay, and these are usually deep in the brain. I'm not going to get into the details with that. Okay, that's higher level neuroscience, so you don't have to worry about that. White matter is going to be composed of myelin. All right, these are going to be axons. Okay, so wherever you see white matter, this means this is where you are having axons. Axons, remember, are going to be or relaying action potentials, right? So this is really going to represent areas of information traveling. Okay, so this is the traveling of information from one part to another part. I'm not so worried about this first point. Okay, corpus callosum. Yeah, you studied it in lab. You looked at it. You know where it is. It is the really the largest white matter tract system in the brain. And that is because this area is responsible for connecting the right and left hemispheres. So this is the area at which the right side of the brain is able to communicate with the left side of the brain and vice versa. Okay, and again, the white color, it's called white matter because we're having myelin there. And myelin, it has a white color because myelin is made of lipid. Okay, it's made of lipid. Okay, another term that we can use to describe white matter is the term tracts. Tracts are bundles of axons in the brain. Okay, and again, it's just another term that we use for bundles of white matter, basically. This next picture is just showing you like the difference between gray matter and white matter. All right, so what you're seeing here is highlighted in orange. On the left here is gray matter. Okay, notice that some of this would be like a nucleus here. Okay, part of the thalamus. And then over here it's highlighting white matter. Meninges. I believe I also mentioned meninges in lab, so you're already familiar with these. These are connective tissue membranes that are going to surround the brain. And there are 3 layers to this, all right? You have the most superficial dura mater. Deep to that, you have arachnoid matter, and then the deepest one is the pia mater. And really the purpose of this is pretty obvious. It's going these meninges are going to help protect the brain, okay? Dura mater is the outermost layer, really, really tough, very strong connective tissue layer. Remember, and again, this was mentioned in lab, that there are these spaces associated with these different meninges. And you should know that the most superficial space, which is just going to be right outside of the dura mater, it's external, it's between the cranium and the dura mater, and this is called the epidural space. It's the most superficial space. Now, remember, this applies not just to the brain, but to the spinal cord as well. All right, these meninges are around both brain and spinal cord. And you've all heard of epidural, right? For example, when someone's in labor, they'll receive a, quote, epidural, unquote. Well, epidural is not the name of a drug. A lot of people think that's the name of a drug. No, this is referring to where they are injecting the drug. Okay, so they're injecting an anesthetic into the epidural space in the spinal cord. And, you know, this is to assure that this anesthetic is going to have a quick effect, all right, because we're right here very close to the vascular network of the brain and spinal cord. So that way our pain responses are going to be blocked much more quickly by injecting it actually into the epidural space. Beneath the dura mater, inferior to it, you find another space called the subdural space. Subdural. All right, so you should definitely be familiar with these spaces, the names of these spaces, both for lab and lecture. All right. And, you know, and I know I mentioned this in person in the lab, and I want you all to think about this. So let's say, you know, clinically, all right, you have someone with an epidural hematoma. That would be a blood clot in the epidural space. Or you have someone with a subdural hematoma. That would be a blood clot in the subdural space. All right, think about the locations here of where these blood clots are. And not just blood clots, but other pathologies such as tumors. All right, you can have an epidural tumor or a subdural tumor. Now, there are different attachment sites for this dura mater, and you've learned about these already. These attachment sites are part of the skull. You remember the sella turcica. The crissagalli is a very important attachment site for the dura mater. So deep to the dura, we have arachnoid matter. It's called that, arachnoid, from spider or spider-like, all right, because it looks like a spider web. So it's called arachnoid matter. Now, just as with dura, you have a space associated with it. Okay, you have a subarachnoid space, which is just beneath the arachnoid matter. And then remember, you have a subdural space that's going to be above the arachnoid matter. All right, now again, think of if someone has a subarachnoid hematoma or a subarachnoid tumor, where do you think is most dangerous to have one of these tumors or blood clots? Do you think it's more dangerous if someone has a epidural hematoma, or is it more dangerous if someone has a subarachnoid hematoma? Well, hopefully you all guessed correctly, subarachnoid. Okay, if someone has a subarachnoid, if there's some kind of mass or blockage in the subarachnoid space, this is much more dangerous because we're closer to the brain tissue itself. Whereas if you have an epidural hematoma, for example, epidural is the most superficial space in the brain above the dura mater. And remember, the dura is really, really tough and strong. It's going to be hard for something to get through there. So epidural hematoma, that's not really that big a deal. I mean, it sounds bad. Or someone says, I have a brain tumor. Someone says, I have brain cancer. I have a tumor in the brain. Well, where is the tumor? That's what it's all about. All right. If you have a tumor, a mass of cells in this epidural space, I mean, any blood clot or mass in the brain is worrisome. But epidural, if it's out there, you just take a bone saw and you get into the cranium and you take it out. It's not that bad. Okay, because it's outside the dura mater. But if we have a mass or blood clot or whatever in the subarachnoid space, which is deeper to the dura, then we're in trouble. Then we want to worry because this is really close to the brain tissue. And a lot of people who have, let's say, a subarachnoid hematoma, they don't even know it. It's too late. The person may die, and it's not discovered that they died from some sort of problem in the subarachnoid space until after death. Once they do the autopsy, they will find this. All right, so subarachnoid problems there are really dangerous. A lot of people just may feel like they have a headache, and you just think it's a headache, and maybe you take some pills for it, over the counter, Advil, whatever. But they don't realize there's actually something much worse going on, and they will die before it ever even gets known. All right. Let's see. Pia mater is the third one. It is the deepest one, and it is directly attached to the brain tissue itself. Very difficult to dissect away from brain tissue. All right. It's very closely attached to the brain. And so because there is no space beneath the pia. And so this is outlining this for you, right? Here you have your cranium, your dura mater, and then you have your arachnoid mater, and very deep you will find your pia mater. Meningitis, all right? So what is meningitis? You've all heard of it. Well, anytime you see that ending itis means inflammation. So it's inflammation of the meninges. Now, where do we see this? What kind of people are we more prone to see this in? And this would be younger, all right? As it says here, a baby, all right, up to like 2 years of age. And of course, we're more likely to see this in this age because, well, obviously these young ones do not have a really strong immune system yet, all right? So they're not able to fight off some of the common bacteria that can actually cause meningitis, all right? Of course, you do have a population of college students too that can get these little outbreaks of meningitis in certain colleges and universities, particularly those living in dorms or close quarters. You may find that. All right, so what happens here? Now we're not going to be able to go through it because you can study this in microbiology if you have to take it. You will definitely study all of the bacteria and viruses that are capable of causing meningitis. There are several of them. And so they're going to get into the central nervous system through the nose and throat typically, okay? And it will go pretty deep, okay? It'll go to the pia mater, arachnoid, and it's going to cause inflammation, right? So we're having swelling, okay? The ventricles, which we're going to talk about, will become enlarged, okay? There will be an increase in the size of the ventricles, and you may have hemorrhage, you may, okay? So hemorrhage means that we will have blood vessels tear and break, and you will have bleeding in the brain. What are some symptoms? So symptoms of this, you should definitely know symptoms of meningitis. The big ones are, okay, well, yeah, you're going to have fever, right? Because we have a bacteria or a virus. So you're going to have fever. Person's going to feel drowsy. They're going to have a very intense headache. Now, one of the big symptoms here, okay, because look, being tired and having a headache, oh gosh, these are really common symptoms, right? I mean, symptoms of all kinds of different things. So really the one that sticks out is the stiff neck, okay? If someone has all of these things, fever, drowsy, intense headache, and they have a stiff neck, okay, this really helps to distinguish meningitis from, other types of issues, okay? The stiff neck is really specific to meningitis. Now, of course, meningitis can be very dangerous, very dangerous. It can lead to death, okay? It just depends on how long this bacteria or virus has been in there, okay? It depends on how long and, the exact kind of bacteria and virus that we're dealing with. Some are more dangerous than others. How do you diagnose it? You're going to take a CSF sample, all right? You're going to do a lumbar puncture where you get a sample of the cerebrospinal fluid. This is called a spinal tap, and you're going to look for bacteria or virus. Okay, so this leads us to a conversation about cerebrospinal fluid and what are called the ventricles, okay? So cerebrospinal fluid, or CSF, is really like the aqueous fluid of the brain and spinal cord, okay? You only find this in the central nervous system. And this fluid is used a lot for diagnosis, all right? If we want to figure out what the levels of something are, or if there's a pathogen present in the brain or spinal cord, we're going to get a sample of CSF and examine that fluid to see what's in there. Okay, now cerebrospinal fluid travels through a specific system of the brain called the ventricular system, ventricles, and we're going to talk about those. First of all, what are the functions of CSF? Well, it serves as a cushion, okay? It provides buoyancy. Like I said, also it's like the major aqueous environment of the brain. It provides buoyancy, okay? So basically, whenever you're moving around, moving your head, the brain actually bounces. It bounces around, all right? So to prevent damage, we have this CSF, okay, to help provide this ability for the brain to sort of, sort of float around or move, with you, rather than just be static and rigid. Okay, so this helps that. It also helps reduce the weight of the brain, okay, because again, the brain is sort of floating around, all right, instead of just being in one rigid position. Protection, yes, okay, protection. Again, this sort of relates to the bouncing around aspect here, right? If you hit your head or the the head moves around. Boxing, right? Boxers, they're always hitting each other in the face, right? I mean, you know, the CSF will help a little bit with protecting the brain from the brain tissue from being damaged from that. And then, of course, chemical stability. Yes, so the CSF is going to carry all kinds of products, all kinds of substances. Like I said, really, the CSF serves as sort of like the aqueous transport and aspect of the brain. You do have your vascular system in there as well that does that, but you also have the CSF that plays a similar role to that. Okay, it's going to help remove waste from the brain. And we're going to have a regulation of the chemical environment through this CSF, all right? And again, we're going to carry signals. We're going to carry all kinds of chemicals, things like neurotransmitters, for example, or just nutrients in general. Okay, so the ventricles. What are the ventricles? These are actually chambers, okay? They're chambers that are going to be filled with cerebrospinal fluid. And they're going to be continuous to one another, continuous. And they go down to the central canal of the spinal cord. So I want to take a look at the arrangement of these. I'm very certain that there will be a question about your understanding of the direction of flow of cerebrospinal fluid through the ventricles. You really need to know this direction of flow. Okay, so let me just jump to a picture. Yeah, okay, that's fine. Let's start with this picture here, okay? So what we're going to start with here is we have these ventricles called the lateral ventricles, and they look like these horn type structures. If you look at this anterior view here on the right, you have two lateral ventricles. You have one in each hemisphere, okay, lateral ventricles. And so the CSF is going to travel here from lateral ventricles. Now remember, we need to know the direction of flow. Okay, so first we have our CSF circulating from the lateral ventricles. Next, that CSF is going to flow to what's called the third ventricle. The third ventricle. Well, now there is another structure here I forgot to mention. So first we have lateral ventricles. Our CSF flows through lateral ventricles and then through this structure called the interventricular foramen. Okay, so inter means between. So we're talking about a structure that's between the ventricles, between the lateral ventricles. And it's called the interventricular foramen. Okay, there's actually going to be a little hole here, okay, that the CSF travels through from the lateral ventricles, and then the CSF flows from the interventricular foramen to the third ventricle. And then from the third ventricle, it goes through the cerebral aqueduct. The cerebral aqueduct is a very thin, very narrow little part of this ventricular system. From the cerebral aqueduct, then, we go to the fourth ventricle, okay, the fourth ventricle. From the fourth ventricle, now the fourth ventricle is right there around the brainstem, where the brainstem begins, okay? And then from the fourth ventricle, we're going down the central canal. And remember, the central canal is that space right in the very middle of the spinal cord, okay? So definitely want to know the direction of flow. Okay, we're going to have from lateral ventricle to the interventricular foramen to the third ventricle to the cerebral aqueduct, very thin little area there, down to the fourth ventricle, and then down to the central canal. Okay, so definitely want to know that direction of flow there. Okay, so I'm just going back to the words that describe what I just said, I believe. All right, and okay, so yeah. So yeah, our ventricles are going to transport CSF. Now, another point here, the ventricles are actually lined by a type of cell we've talked about before, a type of glia. and these would be the ependymal cells. Okay, the ventricles are lined by these ependymal cells, and the ependymal cells are going to have a couple of functions. They're going to make and secrete cerebrospinal fluid. Also, these ependymal cells have cilia, and so that cilia is going to help move along CSF. It's going to help move the CSF through the ventricular system. Right, so we have our paired or two lateral ventricles in each hemisphere. And these are connected by this interventricular foramen. Then we go to our third ventricle, then fourth, and then, or I'm sorry, we go lateral ventricles to interventricular foramen to third ventricle, and then through the cerebral aqueduct. The cerebral aqueduct actually leads to the fourth ventricle, and then we go to the central canal. All right, I've already really described all this on this slide. Okay, so you can read through that if you want some extra repeat there. Okay, the choroid plexus. Now remember, whenever you see the word plexus, we're talking about a collection of either axons or a collection of capillaries. Okay, in this case, we're talking about a cluster or collection of capillaries. And so the choroid plexus is basically going to be found along each ventricle. And it's just, a little blood supply there, okay? Now, also, our ependymal cells are going to be found around these choroid plexes. Okay, and remember, ependymal cells are going to, they're actually going to make and secrete the CSF. They're going to play other more detailed roles in regulating the content of CSF as well. Okay, so ependymal cells really just, in general, you want to associate with, making and maintaining the CSF, as well as the fact that they have these cilia, okay, which help keep that CSF flowing through the ventricular system. Not concerned that you know these numbers, okay, but we do have like a regeneration of our CSF every several hours or so, okay, due to the activity of these ependymal cells. Here's just another picture showing you the flow of cerebrospinal fluid. By no means do you want to learn each one of these steps. You don't need to concern yourself with that, okay? But just so you can sort of have a visual here of the flow of it. And again, what's more important is that you know what I mentioned on previous slides about the flow. This picture is just showing you, look, these are open spaces. So if you look here, you can see the anterior part of the lateral ventricles here, okay, at the front. And you can see the posterior aspect or the caudal aspect of these lateral ventricles. Again, these are in a dissected brain going to look like big empty spaces here, all right, because that's where the CSF is going to flow. And 1 second, guys, give me 1 second. I'll be right back. Actually, like 1 minute. All right, I'm back. Sorry about that. All right, so CSF. This leads us to a little condition. called hydrocephalus. Hydrocephalus. Okay, definitely want to know about this. It can be somewhat common in newborns, all right? So definitely want to be familiar with hydrocephalus. The base word ceph means head. Hydro means water. So literally broken down, this means water in the head, okay? Or water in the brain. So what happens in hydrocephalus is that for some reason or other, all right, there is an obstruction in the flow of CSF, okay? For some reason, CSF is being blocked. It is not flowing through the entire ventricular system. There's a blockage. And so when this happens, you get an increase in the pressure behind the area of blockage, all right? You're going to have an increase in the pressure. And so basically in the newborn, what this is going to cause to happen is the head will actually enlarge, okay? Now let me jump to a picture of this in a newborn. All right, this looks really bad, doesn't it? Okay, this is hydrocephalus in a newborn. We had a blockage in the flow of CSF through the ventricular system, and it resulted in this significant enlargement of this baby's head. Now, what I would like you all to know and understand about hydrocephalus is what is the difference between hydrocephalus in a baby versus an adult? All right, so in a baby, what we're seeing is head enlargement. Why do we see head enlargement in a baby? Because, well, the cranial bones, we've talked about this before, the cranial bones have not yet fused. Okay, remember that it takes a little while for those cranial bones to come together, to meet. And remember that the sutures, you remember the sutures on the skull, you had to identify some of those, that those actually represent areas of fusion of the skull bones, okay, when the bones came together. And remember that, you know, a little baby's going to have these soft spots, right? You've heard of these. And those soft spots are areas where it's just cartilage. Those bones have not fully ossified yet. Okay, so it's just cartilage there, and that's why you want to be careful around the baby's head. All right, but so yeah, this is why we're going to see head enlargement in a baby, because the skull bones are not totally fused yet. So it's possible for all this water that's been backed up to help to actually move out and into this space that's available, right? Now, in an adult, okay, and in the adult, think about this. The adult's brain, or I'm sorry, the adult's cranium is fused, right? It is fused. So think to yourself, are you going to see head enlargement in an adult who has hydrocephalus? No, you are not going to see head enlargement in the adult. All right, so there are a couple of big key differences here then on hydrocephalus in the baby versus the adult. In the baby, you're going to see head enlargement. Why? Because the cranial bones have not yet fused. In the adult, you are not going to see head enlargement because the cranial bones have fused. So think to yourself, what's more dangerous? Now, is hydrocephalus going to be more dangerous in a baby or an adult? All right, think about this. In the baby, it's very obvious. You can see this. All right, you know that baby has hydrocephalus. That's what they've got. There's no other option. All right. If they're having a large head like this, yes, it's hydrocephalus. So therefore, it's really easy to diagnose, right? You can see it very clearly. It's obvious. So then what do? You just go in and you drain that fluid out. All right, you drain the fluid. Now, what if it's an adult? If it's an adult, you don't see this, do you? So it's much more hydrocephalus is much more dangerous at the end of the day in a grown person versus an adult just because of the fact that you don't see this. So the person, again, like kind of like what I was mentioning with meningitis, or something I was mentioning, that, you know, the adult is going to be like, my head hurts, I have a headache. That's going to be their symptom. They've got a headache. That could be so many things, right? When you go into the clinic or hospital, doctor, whatever, when you say you have a headache, you're not always given any kind of a brain scan right away. Okay, that's not always done. All right. So the point is, the adult, you don't know it. You can't diagnose it as early. So it's much more dangerous in the adult. Let me go back here and say a little more about it. Okay. So again, you know, in the adult, what we're going to see is, well, we're not going to see head enlargement. And the reason this is dangerous is because we're going to end up in this adult that we're going to end up compressing blood vessels due to that higher pressure, okay, due to some sort of blockage in the CSF, the ventricular system. So that increased pressure from that CSF buildup is going to place pressure on surrounding blood vessels. And then if that happens, I'm sure you can imagine the rest of the story, right? If that happens, then we're reducing or blocking blood flow to tissue, and that can cause the death of brain tissue. And if this is continuing to go on, the person can have very serious problems. They can end up with brain damage, okay? So this is another result here. In the newborn, very unlikely that you're going to end up with brain damage because you see it right away. And what do? You're just going to drain the CSF. There's something called a ventricular shunt that you can insert there, and you're going to drain that CSF out. But in the adult, again, you know, you don't see it, so it's not diagnosed as early, most of the time, and you end up having a prolonged period of compression of blood vessels, and in this case, you would be more likely to see brain damage in the adult compared to newborn. Another point I want to make, and I did not mention this in the slide, most common area for blockage is going to be the cerebral aqueduct. All right, you might as well know that. I don't know if I'm going to ask you about it. It might be a question. So, you know, you should take note that the area where this is typically going to happen is going to be the cerebral aqueduct. And let me just go back real quick and show you that. Look at this ventricular system. Our cerebral aqueduct is really narrow, so it's much easier to get some sort of blockage go on here in the CSF, and in the cerebral aqueduct. Okay, so that's the most common location for a blockage. All right, so this is where I can start skipping slides a little bit because you know about the anatomy here. Cerebrum. So we're starting here with the cerebrum. Remember, cerebrum, you have two hemispheres. Really accounts for a large part of brain mass. Okay, the amount of space there, a lot of it is actually cerebrum. And again, you know, cortex is out here. So we're dealing with things like, functionally, we're dealing with things like memory, all right, thought processing, judgment. Okay, you also have termination of sensory perception happening out here. You also have the initiation of motor activity happening from here. Okay, so remember, all sensation comes from the body, goes up to the spinal cord, and terminates in the brain. Okay, specifically, it's going to terminate in the cortex. Whereas motor activity goes the opposite direction, right? Motor activity, especially for voluntary motor activity, is going to start at the cortex and then go all the way down to the spinal cord, out to the body. Surface markings, you already know that from lab. Okay, again, just another reminder of brain anatomy. You can look at that. Another reminder here, your lobes, remember the lobes. All right, so we have 4 lobes. And again, you've done this in lab already. But what we're going to do here is detail these out a little bit in terms of function. All right, so central sulcus, you remember the central sulcus. Let me just show you that on the next slide as a reminder. You have this central sulcus, and remember that a sulcus it's not associated with any function at all right? There's no function associated with a sulcus. A sulcus is a line, okay, that separates gyri. Because so the brain, throughout evolution, the size of the brain got larger, okay? As humans evolved, the entire size of the brain got larger. But, the size of the cranium itself didn't grow as large. So we had all of this new tissue and evolving brain tissue. So it's like it didn't have a whole lot of room to move outwards. So it got squished. Okay, so that's why we have all these convolutions. These are all convolutions, gyri, okay, a gyrus. All these gyri are convolutions. It's like the brain tissue sort of folded in on itself, okay? It's sort of bunched up, okay? So that's how our sulci were created, okay? It was due to this brain tissue sort of folding in on itself because it was growing in size, okay? So again, sulcus, you don't have a function for a sulcus. But for a gyrus, right, that's the actual tissue. That's the actual brain tissue. And yes, each one of these gyri have a specific name, and each one of them have a specific function. But I'm only going to ask you to know 2 of them. The precentral gyrus and the postcentral gyrus. Those are the gyri that for lecture, you do want to know what the functions of these are. Okay, so structurally, anatomically, we have a precentral gyrus located anterior to the central sulcus, and we have a postcentral gyrus located posterior to the central sulcus. Okay, so the central sulcus is a landmark. It's a landmark for us to help us distinguish the difference between the precentral and the postcentral gyrus. All right, so what are the functions of these 2 gyri? You should definitely know. I'm sure there'll be a question on it. So the precentral gyrus is associated with initiating motor commands. Okay, so this is the spot for where motor commands start. Now, I mean, it's actually a lot more complicated than that. There's actually a part anterior to the precentral gyrus, which I am going to mention later, that is associated with the really initial parts of gathering information on what kind of movement should we direct to the precentral gyrus. Okay, but you should understand that the full motor command is actually going to be integrated and set forth from the precentral gyrus. The postcentral gyrus is the one associated with sensory. So postcentral gyrus is the final spot for incoming sensory input. Now, of course, sensory information is going to travel through all sorts of places before it gets to the postcentral gyrus. Okay, but our sensory information in general, once it goes through all these lower parts, is going to end up synapsing or terminating there in the postcentral gyrus. Now, similar to what I mentioned about the precentral gyrus, the postcentral gyrus, this information getting there, synapsing there, I mean, we say it terminates there, but understand that the sensory information that we say terminates there is actually going to branch out to other parts of the brain, okay, for us to get a total understanding of whatever sensation is going on, okay? But yeah, in general, you know, precentral gyrus, postcentral gyrus, they're major locations, okay, for outgoing motor information and incoming sensory information, respectively. The rest of this is just anatomy, other anatomy. Here's a reminder of your anatomy of your lobes. Remember the lobes. All right. So, of course, the lobes have specific functions. All right. And we need to know about these functions. Okay. So, the frontal lobe is really the most interesting lobe of the four. And I will spend a little bit more time talking about this frontal lobe. Okay, it is rostral. Okay, it's in front. You know where the frontal lobe is. What are some functions? Now, there are a lot of functions going on here. You do have voluntary motor function going on here. So basically, before the precentral gyrus sends out a motor command, there's going to be an area in the frontal lobe which sends input to that precentral gyrus about what kind of voluntary motion to do. Other functions of the frontal lobe. Now, there are actually a lot more functions of the frontal lobe than what are listed here. And you can get really detailed about it, okay? But things like motivation, foresight, planning, okay, decision making, okay, the ability to use strategy to plan something. Decision making is a big deal here in the frontal lobe. The ability to reason, all right, so the ability to reason, to use logic and reasoning to do things like plan activities or to plan what you're going to say, to plan what you're going to do, to plan what action you're going to take next, to plan what behavior you're going to do next, all right? Memory. Now, memory is located in a variety of parts of the brain. The real core of memory is not in the frontal lobe. The most primitive core aspect of memory is actually located in a brain region called the hippocampus. But you do have a lot of memory networks going on in the frontal lobe because you have to have access. The frontal lobe has to have access to memories in order to make proper decisions about planning things, okay? You have to have access to your memories in order to have personality, to have motivation, all right? Personality is a big deal in the frontal lobe. Okay, like I said, there's a lot more listed here that's going on. Okay, you know, something that I don't have specifically written out here is decision making, all right? Decision making is a big deal there in the frontal lobe. Personality is a big deal in the frontal lobe. All right, but and that relates to social judgment, right? It relates to this other one, very important, inhibition. Okay, the ability to censor your behavior is located in the frontal lobe. That is really important, and I want to expand on that just a minute and sort of tie a lot of these functions together here. Okay, like for example, the types of judgments or decisions that you make in social situations and how that relates to the ability to inhibit your behavior. Now, a lot of this you learn in psychology, in your psychology class, okay? But from a neurological perspective here, all right, frontal lobe, we're going to have these. Now, I'm going to go into this in more detail later, but I'll go ahead and sort of preempt it. Let's say you have damage to the frontal lobe. If you have damage to the frontal lobe, and again, you know, keep this in mind, you have to know the functions of the brain regions because if you have damage to that part, then that means that's what function will be lost. That's what you will be missing, all right? That's what you will no longer have. If you have a lesion or some sort of brain damage to this part, you are no longer going to be able to do these things. So, certain things. Okay, so for example, prisoners, all right, hardcore prisoners. I'm talking about like death row inmates. people who have done very serious crimes, murders, that kind of stuff. It used to be, back in the day, that science could use prisoners as guinea pigs, basically, all right? Science was allowed to take these prisoners and study their brains, all right? We're really not, it's, not really doing that anymore these days, but a lot of valuable information was achieved from those experiments, a lot of information. And what was found was that in very hardcore criminals, people that did really bad things, they had smaller frontal lobes. Smaller frontal lobes. Now, you can take that a couple of different ways, okay? You can wonder to yourself, well, does that mean that this person was born with a small frontal lobe? Did this person have a frontal lobe that did not fully develop? Okay, that's one way. Second way is, or later in life, was there some sort of damage to the frontal lobe? Okay. In either case, you're going to have deficits in these things. So, it comes to follow. It's like, well, these people who did these horrible, horrible things on death row, they weren't able to make the proper decisions socially. They were not able to stop their behavior. This is a key word here, behavioral inhibition. If you have a problem in the frontal lobe, you are not able to control your behavior. You don't have the ability to do it because the frontal lobe is like the check. The frontal lobe is your ability to reason and logic and, like, is this the right thing to do or not? And if you'll know if it's the right thing to do. If you know it's not the right thing to do, then you don't do it. That's because your frontal lobe's working. If your frontal lobe's not working, you're not able to stop yourself. Okay, so this really led to a lot of really interesting information about, which led to for other controversies, such as, are these people on death row, for example, are they at fault? Is it their fault? Kind of crazy to think about. All right. I'm not saying I have an opinion either way. I'm just talking about, the science here on it. All right. So, frontal lobe, really interesting part of the brain. Another thing I'm going to go ahead and mention about frontal lobe. All right. So drinking alcohol, right? Alcoholic beverages. You may remember that alcohol binds to GABA receptors. You remember GABA. GABA is the major inhibitory neurotransmitter of the brain. Well, we have a lot of GABA receptors, a high density of GABA receptors located in the frontal lobe. So when a person is consuming alcohol, it's the frontal lobe that is the first part of the brain to be affected. So what happens is the alcohol binds to the GABA receptors in the frontal lobe, inhibiting your ability to do some of these things. Okay? Your judgments start to fail. All right? You start to not inhibit your behavior anymore. You will start to do things that you wouldn't normally do. All right? Okay, things like emotion. All right, now you can have differential effects here. It says here aggression. Okay, aggression, again, you must understand that aggression is actually a more of a core function in another part of the brain called the amygdala. Okay, the amygdala is a part of the brain that really is responsible for aggression. It's just that the frontal lobe is associated with whether or not we're going to act out our thoughts of aggression. Are we actually going to express that aggression? That's the frontal lobe. Whether or not we are experiencing aggression on the inside is associated with amygdala. Okay, but the actual acting out, the decision to act out aggressively is the frontal lobe. Okay, I'm just going on about the frontal lobe. But, you know, it's just such an important part of the brain. It does so much, and, you know, you can really tell, like, if somebody has frontal lobe damage. You can tell. All right, so that point about alcohol, though, I do want to mention. I want to expand on that. So, yeah, you know, alcohol is first going to affect the frontal lobe due to the high density of GABA receptors there. And so, yeah, you will see very, you know, differences in function here of all these things. Now, once a person has had all of their GABA receptors in the frontal lobe saturated, so let's say a person is continuing to drink. They continue to drink. Well, at that point, all the GABA receptors in the frontal lobe are occupied. Okay, so you're past this point. The second place the alcohol is going to go is to the cerebellum. I'll talk about the cerebellum and its role in motor coordination. Okay, now remember in lab 10, lab 10 about sensory systems, there was a test there done to test a person's proprioception. It's the same test that police officers use when they suspect someone is driving under the influence, right? Same test. Well, that they're measuring the cerebellum is what they're doing, all right? Because cerebellum is associated with motor coordination and proprioception. So that's how you can really tell. Look, if that person is failing these tests that are designed to measure function of the cerebellum, they're definitely drunk because they've already done saturated up the frontal lobe. Because after the frontal lobe, then we start to see cerebellar effects. Now, let's say a person is continuing to drink, and then we have all of the GABA receptors in the cerebellum saturated. Well, the alcohol's got to go somewhere in the brain. So the last place it's going to go is the hippocampus. The hippocampus is associated with learning and memory. Okay, so if it gets to a point where the person is drinking so much that the hippocampus starts to be affected, that's when they have blackouts. All right, that's when a person will just pass out. They will have blackouts, or they will have total loss of memory of what happened during the time they were drinking. Now, in severe alcoholics who get to this point of drunkenness commonly, very frequently, what ends up happening is they destroy their hippocampus and they end up having amnesia. It's called Korsakoff syndrome. And they will forget everything they've learned. They will forget how to read and write. They forget who they are. They forget people they know. They have total amnesia. And then you actually have to retrain these people how to do all of these things. It's a really sad situation. All right, I have gone enough on that tangent. The next lobe is the parietal lobe. And all I really want to say about this is parietal lobe has a lot to do with sensory systems, okay? Parietal lobe is going to integrate, as it says here, your senses, okay? So we talk about some of your visual information is processed there. Taste, smell, all right? All of your senses really are going to be associated with being integrated or analyzed in the parietal lobe. Occipital lobe is all about vision, Okay, this is called it's considered the primary visual center of the brain, occipital lobe. And this is interesting. Think about this. If occipital lobe is all the way in the back of the brain, right? It's all the way caudal. Where are your eyes? Your eyes are all the way in front, right? So why would our visual processing center be all the way in the back of the brain? Why would it be the farthest away from the eyes? Why wouldn't the primary visual center be right near the eyes? Well, the reason for this is that all of the visual information that's coming in through the eyes, all of the axons, okay, bringing that information in, must travel through other parts of the brain to pick up information relating to things like memory and learning, things like emotion, all right? We have to have input to our visual processing relating to all of these things. Like, do I remember seeing that? Do I have an emotion or a feeling about what I just saw? Okay. You have to have a lot of other input in deeper areas of the brain that are added to that visual stimulus before that final analysis or that final version of what you're seeing is totally processed. So that's why the primary visual center is in the back of the brain. All right. We have to have time. We have to have, which I shouldn't say time, but we have to have those axons from the eye go through other parts of the brain before we can make our final decision in the back of the brain about what's going on with that visual stimulus. And think how fast this happens. Are you thinking about this? Nope. Temporal lobe. Temporal lobe is really mostly about hearing. Okay, now there are some other things going on there, but temporal lobe is all auditory, a lot of auditory going on there, right? Functions in hearing. Now, learning and memory is also in part of the temporal lobe, and specifically that would be in that very special brain region that I mentioned called the hippocampus, which we'll bring up again. Okay, hippocampus is located in just part of the temporal lobe. And then, we do have some other functions shared over there as well. And you can note those, all right? But our big deals are really hearing. Okay, so this is a pretty good little picture here because it shows the different lobes and sort of details out the different functions, okay? All right, cerebral cortex. All right, so remember the cortex is really the, well, here it says the executive control of the brain. Okay, this is where intelligence is. This is also where consciousness is located. It is consciousness, a very controversial, difficult subject that still really not a great deal is known about. Okay, but we have through PET scans, okay, through PET scans as well as MRIs, been able to locate certain aspects of consciousness, and it has been found in the cortex. And again, I've already detailed some of this out, right? In the cortex, we do find our postcentral gyrus, which is associated with sensory. We have our precentral gyrus, which is associated with motor. All right, and then all the other things, functions I mentioned about the lobes, all right, because the lobes, the outer part of the lobes is cortex. So all of those functions apply, okay, to the cortex. Pretty large part of the brain cortex. Now, cerebrum is 83% of the brain, and 40% of total mass of the brain is actually cortex specifically. Now, we're going to use PETs, MRIs, to look at brain activity, okay? And we can sort of detail out specific parts of the cortex that do specific functions. And when we're able to do that, we call those specific parts domains. Now, also, higher functions in the cortex, okay, we can call association areas. And there are very, very, very many of these association areas. I am going to mention a few of them, but we certainly will not go through all of them. When we say association area, we're talking about an area of the cortex that does the highest level or the highest possible level of analysis of something, okay? It's the highest level of analysis of any kind of function, okay? Whether that be memory, sensory, like vision, auditory, that kind of thing. We have auditory association areas. We have visual association areas, etc. And so this is just a picture here of an MRI. It's actually an fMRI, functional MRI, which we really use for looking at, okay, if we want to see what parts of the brain are functioning during certain tasks. Okay, so as we can see with C, now we're looking at the areas that are in red. That indicates area of higher activity. So when we're looking at something, what's going on? Occipital lobe back here is lighting up. Hearing, when we're hearing, temporal lobe is lighting up. Speaking, now we'll talk about speaking a little bit later on. Speaking is going to be located here, sort of parieto-temporal, sort of parieto and also temporal, okay? Speaking. Thinking, look at this. Now, you know, this little image is very not specific. This is really not giving us any information, okay? Because, well, what is the person thinking about, right? What are they thinking about? Okay, if they're thinking about eating or drinking, guess what? It's going to be really red right about down here in the hypothalamus. Okay, just for example. So we don't know what this person with this image, what were they thinking about? Okay, but what you can conclude from this image is that thinking in general, thinking about anything, is going to involve, it will involve a number of areas of the cortex, because when you're thinking, it's like everything's kind of coming together. So you'll see lots of different parts of the cortex light up. But again, it will depend on exactly what you're thinking of. All right, guys, you know what? I think I'm going to go ahead and end this video here. I think that's good for now. So it looks like I'll be making 2 more videos on this PowerPoint. Okay, so take all that in, and I'll get you your next video up as soon as possible. Have a great day.